Francis Awino. Francis Awino works with the Nairobi Hospital. He has a specialty in hematology and blood transfusion. And today he will take us through immunohematology and transfusion reactions, plus any disorders arising from blood transfusions and even organ transplants. So welcome, Francis. If you're there, you can project, you can share your screen. Francis, are you there? Francis, you can unmute.
Hello, you are getting me?
Yes, we can hear you clearly.
Are you able to see my screen?
No.
Okay, yes, I will. Okay, damn it.
Yeah, as he's sorting that, we welcome those who have joined. I see the attendees are rising. The numbers are coming up. Welcome. Francis, we are still unable to see your screen. Let us be patient as our presenter is sorting his slides.
Okay.
Francis, are you able to share the screen?
Are you able to see it, Bruce?
Good, good. Thank you. Yeah, you can. We are able to see it, so you can make it on the slide. Make it on the slideshow. Yes. Okay.
That's okay now.
Yeah, it's okay. So welcome, Francis. We had already introduced you, so you can proceed.
Good evening, everyone. This is Francis Awino, a medical laboratory officer, hold a bachelor degree on the same from Maseno University. Currently specializing in hematology and transfusion from JKUAT, a medical laboratory officer practicing at the Nairobi Hospital. In today's session, I welcome every one of you. It will be a learning opportunity. It's also a new area, and due to the emerging diseases and also advancement in technology, I'm sure in future we'll have more transplantation centers coming up. Currently, I know the top tier hospitals in Kenya, like the Kenyatta National Hospital, even here at Nairobi Hospital, Aga Khan, some of them, they have already kickstarted the transplantation process, and this is something that is going on. Besides us not having the transplantation center, I'm very sure we also offer post-transplant care, because we have several patients who maybe were transplanted in India or in other countries, but now the follow-up is done locally. So welcome all. We are going to, this is a branch of transplantation in immunohaematology, and today we'll be looking at graft rejection, which determines the success of a transplant at the end of the day. That's okay, Bruce, so we can go ahead. Hello, Bruce.
Yes, you can proceed. It's okay.
So the objectives for today's presentation, graft rejection is such a wide area and something that we cannot exhaust in one presentation. And we'll alliance with Bruce and the concerned team so that we have another second presentation where we can continue with what we will discuss today. So today we look at the mechanism of graft rejection. So of key interest is the recognition of antigen, because that is where it starts. So once we are able to see how the graft antigens are recognized by the host immune cells, or the host immune cells like the T cells, then we'll be able to understand the subsequent activities and how they unfold. And then the next one is to describe the mechanism of T cell activation. How does the host T cells gets activated once this foreign organ or maybe a tissue has been transplanted to a recipient that direly needs them? And then finally, to just sensitize laboratory personnel on graft rejection. As I've said on the introduction, there is emerging diseases. Let's say kidney kidney failure, those ones might require a kidney transplant. And even those ones who are suffering from hematological malignancies, the likes of leukemia, the best solution is stem cell transplant. So we'll see more and more of these cases coming up. And with the advancement of technology, we are looking forward into a future where in Kenya, other people can travel far to seek this essential service. So as the success of transplantation relies So the success of transplantation is limited by the immune response of the recipient to the donor tissue. Remember, we are introducing a foreign material to the recipient. The recipient here is the patient, then now the donor. The donor is an healthy living person, so first that will be the requirement that they must be in their full health of state, so that we don't, it is of the maximum benefit to both the recipient and the donor. You are taking someone's kidney and transplant to somebody else with a kidney that is not functioning. So what we have to ensure that the donor must be able to function on the remaining one kidney. So rejection, therefore, is the recognition of the allograft and immune response against the allograft by the recipient immune system, resulting in allograft destruction. So in rejections, what happens is that once the tissue has been transplanted or the recipient have received the foreign graft from an allogeneic donor, they will mount an immune response. So there is also several process that happens before somebody is transplanted. You have to prepare the recipient to receive the new graft that is coming in, and this one includes immunosuppression. So we are just reducing the level of activities that is happening. The level of the immunological reaction, so that the body or the immune system of the recipient is able to tolerate the coming graft. But sometimes that is not the case, and you find that instead it elicits a very strong immune reactions that end up destroying the newly transplanted tissue. So to an extent that it is not grafting, and at the end of the day, we might even have to remove the implanted tissue to save the recipient. So that is now the rejection. So if such happens, then now we have not achieved the goal of transplant. So it is an adaptive immune response because it involves the cellular mediated immune response. Mostly the cells that play key roles are the lymphocytes, and the lymphocytes here are the T cells and the B cells, where the B cells will place the key roles in the humoral immune response by producing antibodies attacking the grafts, and the T cells will employ the likes of the alpha T cells, the CD4s, and the cytotoxic T cells, the CD8. So what is an alloantigen? Alloantigens are molecules on allograft that are recognized as foreign. Remember, it is a whole tissue. It could be a kidney, it could be a liver transplant, or even a skin transplant. But anyway, most of the time it would be either stem cell, or you are dealing with especially most people who have been transplanted around the renal failure. So kidney transplant. So those the molecules on allografts, or the tissues of the allograft, that is the organ itself, that is what is being recognized as foreign by the recipient immune system, and that's what we refer to as alloantigens. So a case where you transplant someone with, let's say, an animal tissue, or maybe we are doing a study and we want to investigate something, then you take an human tissue and you transplant in a mice. That is a xenoantigen, because they are of different species. In alloantigen is of the same species, and then xenoantigen, we are talking about molecules on xenografts that are recognized as foreign. The lymphocytes and antibodies that react with alloantigens or xenoantigens are described as either alloreactive or xenoreactive, respectively. So if it is a xenoantigen, and now we have antibodies attacking the animal tissue, we call the cells xenoreactive, or the antibodies xenoreactive. If it is of the same species, that is from a donor, then we refer to them as alloreactive. So allogeneic transplantation is far more commonly practiced and better understood than xenogenic. So mostly xenogenic will apply in research setup. In research setup, most of the time when you want to maybe investigate something, then you would want to use an animal, let's say the lab animals, like you can use the mice, or even the monkeys, the baboons, and all that. Then you place the tissue, and even understand the mechanism of graft rejection. You want to kind of stimulate the T cells and see how they are going to react. So that's where you might need to do xenograft. But as in the clinical setup, what we'll be seeing is alloantigens or allograft, because we will be receiving live donors, that is human donors donating to fellow humans. And then rejection starts with the placement of the graft when the immune system of the host detects the foreign graft tissues. So immediately the graft is placed, or immediately the patient is taken to theater, and then the new tissue has been the surgery is successful and the whole thing has been done, in that I've got a successful transplant, the recipient immune system will recognize that as a what? As a foreign material, and it will start to launch an attack to the foreign material immediately. So the strength of that reaction is what will determine whether that tissue will be totally rejected or not. So almost immediately, donor antigens enter the systemic circulation and the lymphatic system back. So remember, these are organs like the kidneys. They still have blood running through them. They have cells like the lymphocyte cells are there, the white cells, the red cells, and all that. So immediately, then the surgeons have anastomosed, which means they have sutured, joining these blood vessels of the recipient to that of the donor graft. There will be circulation or flow. So these graft antigens or the alloantigens that have been introduced in the system will try to traverse the recipient system by accessing the lymphoid or the secondary immune organs. And from there, they are exposed to the recipient immune cells like the T cells, and that is where the reaction takes. That is where the reaction stands. So by accessing the secondary lymphoid system, that is mostly the lymph nodes and the spleen, the naive T cells will be activated, and there are only two pathways that we can. The major two pathways that activate graft rejection, one is direct and another one is indirect allorecognition. So as we proceed with the presentation, you will see what happens in the allorecognition pathway, like how does the direct pathway occurs and how does the indirect pathway occurs, and also the semi-direct pathway. So the stages, one will be the sensitization stage and then the effector stage. Sensitization stage is all about recognizing and activating the T cells. In the effector stage, that's more of clonal expansion of the T cells. We are producing mature T cells which will be able to recognize the graft antigen and mount very strong immune reactions or rejection to this particular foreign antigens. Then clinically, it will present in three stages, that is hyperacute. In our next presentation, we'll see how the effector stage look like, and we'll also see how the clinical manifestation look like. So in hyperacute, it's more of immoral. It's not really mediated by the T cells. So hyperacute rejection is based on already existing antibodies. So the patient could have been sickler. The patient could have been suffering from hematological malignancy, and maybe they have received multiple transfusions. And through these transfusions, they have developed some anti-HLA in their system. So those antibodies will be able to mount an immediate immune reaction by attaching themselves on the endothelial lining of the newly implanted graft. And by doing that, there will be occlusion of the new graft and finally rejection. And then acute rejection phase, that one is mediated mostly by the direct pathway, which is the cytotoxic T cells. The CD8 positive cells are the ones that plays the major role there. And then finally, the chronic rejection is mostly mediated by the T helper cells. And this one you will see is how it happens is that there will be overproliferation of the vessels, like mostly the arterial occlusions. The cells are activated in a way that they produce tumor growth factors, and that one enhance the growth of or the thickening of the wall of the arterial. And then finally, there is occlusion, and blood cannot go through to nourish the newly planted graft, and it gets rejected. That one can happen from 6 months post-transplant. For acute, it can happen within 7 days, 1 week, but hyperacute is in a matter of minutes, in a matter of hours, because remember, it is already existing antibody, so it starts immediately. So recognition of alloantigen, immune responses to allograft, where alloantigens will elicit both cell-mediated and immoral response. As we have said there, in the hyperacute phase, you've seen, we talk about the antibodies, that is immoral. And then now in the acute and the chronic phase, we'll be talking about the cellular mediated, the CD8 and the CD4. Recognition of transplanted cells are self or foreign is determined by polymorphic genes. So there is major histocompatibility complex is the one that plays the key role in graft rejection, because it's the one that determines, in MHC, we'll be able to determine whether the host immune system is trained in a way that they know the self-MHC and the non-self-MHC. So if you introduce the donor MHC in a recipient system, the naive T cells will be able to recognize that this is a non-self, and then they mount an immune response towards that. And this major histocompatibility complex, they are inherited as an haplotype, that is a group. It's not a single gene, like we have got class 1 and class 2. So in class 1, there are like 3, A, B, C. That is MHC class 1, we have got HLA-A, HLA-B, HLA-C. And then class 2, we have got HLA-DR, HLA-DP, HLA-DQ. And it is inherited that way, like it is a whole haplotype, and then they are expressed codominantly, and you inherit that from both parents. These genes are highly polymorphic. So I have already started introducing this. That is the major histocompatibility complex. As we have said, that the cluster of genes that encodes for them. This MHC, they encode for the glycoproteins. So these glycoproteins are membrane receptors, and they are found on surfaces of T cells. And also, like in class 1, you find them in almost all nucleated cells. Then we have got MHC class 2, which are mostly only found in antigen-presenting cells. That is macrophages, dendritics, and also the B cells. Then the human MHC, we talk about the HLA, that is the human leukocyte antigen gene complex, and they are located on chromosome 6. So that diagram just shows you the types of HLA that we have. So HLA, like for example, class 2, I said we have DP, DQ, and DR, and then class 1, we have got A, B, and C. Class 3 mostly complements and other cytokines like tumor necrotic factors, but class 3 does not play a role in antigen presentation or antigen processing, but they play a role in immune system. Like now, you know, the complement system, the CT, the C4A, the C4, if you follow the classical pathway, the end product is membrane attack complex that introduce lysis, and that is how they are able to destroy the infected cells through that pathway. Recognition of alloantigen. The MHC complex is the locus of polymorphic genes that encode the molecular targets of allograft rejection, where the MHC molecules are responsible for all strong or in bracket rapid rejection mechanisms. And allogenic MHC molecules are presented for recognition by T cells of a graft recipient in 2 different ways. So the first one is the direct presentation, and then the second one is indirect. We also have a third one, semi-direct presentation, but it's more of a blend of the two, of a direct presentation and indirect presentation. So I'll talk about it also in brief after seeing what the direct presentation is and the indirect presentation. So what happens in direct presentation is that the T cell recognition of an intact MHC molecule displayed by the donor APC in the graph. So here, the donor antigen presenting cells like the dendricyte, like the dendritic cells, they interact directly with the antigen, the recipient antigen presenting cells. Remember, these are organs, as we had said. So within the kidney, there is blood. And within that blood, as much as we have got cells, we have got the endothelial cells lining the graft. We have also got the blood that nourishes those particular cells. And inside that blood, we have got the white cells, which are the T cells now. And these T cells, we also have the dendritic cells, which are the professional antigen presenting cells, the macrophages, and the B cells. So in direct presentation, the naive T cell of the recipient is able to recognize and interact directly with the donor MHC. So this is where the donor APC, so like, as you can see in the diagram here, we have got the allogenic antigen presenting cells in the graph, and that is the dendritic cells. and we have got the alloreactive T cell from the recipient and is able to bind. Remember, antigen presenting cells, they have got MHC class 1 on their surface and also MHC class 2. MHC class 1 is present in all nucleated cells. And so in direct allorecognition, what happens there? What happens is that the T cells, and here we're talking about the CD8, the cytotoxic T cells, they are able to bind directly to the MHC class 1 displayed by the donor APCs. And by doing this, they get activated, and once activated, they release things like once the T cells becomes activated, there is now the proliferation. So they will grow like in numbers, and they are able to differentiate and able to recognize this direct allo-MHC molecule from the graft. And by doing that, we have got effector T cells coming in as a result of this activation. So the effector T cells, what they do, they will release things like perforins, which are able to create pores on the graft tissues. And by doing that, causing much destruction to the newly planted graft. And then in the indirect presentation, that is the indirect pathway, the recipient APCs process the donor peptides and engage the recipient lymphocytes. So now what happens here under the indirect pathway is that the donor or the haloantigens are processed by the recipient antigen presenting cells. The dendritic cells are able to engulf or rather do phagocytosis or even endocytosis. So through the endocytic pathway, once this one has been digested, they are broken down by proteasomes into peptides. And then now they are broken down by the lytic enzymes. And then once they're broken down by the lytic enzymes into the peptides are presented on the surface of MHC, of the MHC class 2. And then the MHC class 2 is able to display this to the recipient T cells. So mostly the CD4 cells will be able to recognize the processed donor allograft and mount a very strong immune response that in turn destroy the donor tissues. How does this one happen? Because remember, CD4 are helper cells, so they'll be able to differentiate, they'll be able to, that activation, they'll be able to release things like interleukin-2, and then interleukin-2 will recruit more cytotoxic T cells, which will be able to destroy the donor tissues. They'll release cytokines like the interleukin-4 and 5 that will be able to activate the B cell line that will differentiate into plasma cells. And then now the plasma cells differentiating into the antibodies that are allospecific to the transplanted tissues. And these antibodies just mount again another strong immune response. So that is how the indirect allorecognition pathway takes place. So as you can see in the diagram, you are seeing there is an uptake of the allogenic MHC, and then this one goes through antigen processing. If you remember the immunology, how the antigen processing takes place. And then once the allo MHC has been processed, the peptides will be displayed on the surface of MHC class 2. And then this will be recognized by the recipient lymphocytes, and in this case, the CD4 positive cells. So mostly indirect allorecognitions will play much role in chronic, in the clinical presentation, it will attribute to the chronic phase, while the one we saw earlier, direct presentation will result into an acute rejection. Hyperacute, we said there is no cell mediated, it is existing antibody. So just to tie the two, because in this lecture we will not cover on the clinical stages, but we just want to see how the sensitization takes place. So now the semi-direct pathway, the same recipient APC activate both the direct and indirect pathway. So what happens here is that we are at the lymphoid organ. So this is a secondary lymphoid tissue. Remember, once the surgeon have anastomosed and we have joined the vessels, we have joined the vessels of the recipient to that of the renal vessels from the allograft. It becomes one, and the circulation has to continue. So as this happens, or even the lymphatic system, all the systems are activated. So you'll find that the cells will move along the systems, and now we have got now the allograft or the haloantigens. They are moving to the secondary lymphoid organs. I mean, to the secondary immune organs such as the lymphoid. And here we have got naive T cells. So these T cells, instead of because remember the dendritic cells, the resident antigen presenting cells in the recipients will possess both MHC class 1 and MHC class 2 on their surface. So here we'll have MHC class 1 interacting directly with the the donor antigen presenting cells, or rather the donor MHC molecule. And then we, at the same time, the antigen presenting cells like macrophages in the reticuloendothelial system, in most cases, what they will do, they'll be able to engulf, at the same time, be able to engulf or phagocytose the donor haloantigens. And once this one has been phagocytosed, it will be processed and displayed on MHC class 2. And that will activate the CD4, the recipient CD4 lymphocytes. And then we are having one antigen presenting cell activating both pathways, both the indirect and the direct. And that's so that is the pathway that now form the semi-direct pathway. So it mounts a very strong immune system, and we'll be having more of these T cells, the cytotoxic T cells on the other hand proliferating, and we also have the helper T cells proliferating, and then they become effector T cells, which can now migrate or be transported. They find their way to the organ, and once they find their way to the newly implanted organ, let's say the kidney, they mount a very strong immune rejection, and that organ suffer slow death. So if it was transplanted, for that case, if it was a kidney patient, you are doing a follow-up maybe 2 weeks after transplant, or even a month, let's say 2 weeks or even a month after transplant, and nothing is correcting. The creatinine is still very high, and there's still signs. So that one is telling you that the new kidney, of course, you don't expect everything to correct, because remember it is engrafting to correct immediately, but at least the new kidney should have started functioning. So this patient, as much as they are having derailed kidney function test, but the picture should be different from what was there pre-transplant. But in such a case, you'll find that there is no really much improvement in terms of the markers that we are using to follow up. If it was a kidney chronic patient, I mean, it was a renal failure patient, then you would find that doctor would still need dialysis or something of that sort. Yet we have done transplant, then it means that newly transplanted organ is not functioning. And the reason being, again, because there's a very strong inflammatory reactions going on. There'll be very high fever, things like that. So there is those signs that you'll be able to see that there is a rejection that is going on. Because there's immunological reaction will incite even fever and even a lot of pain for the recipient. So really the symptoms are getting worse instead of improving. So that's now the rejection phase. So in this diagram is a summary of what direct halo recognitions look like and what indirect halo recognition look like. So as we have said, in direct halo recognition, we are having the donor lymphocytes, in this case the CD8, they are able to interact directly with the recipient lymphocyte, the CD8, they're able to interact directly with the donor APC, that is the donor antigen presenting cells, where they bind the MHC class 1 direct, and then from there they get activated and differentiate and produce more effector cells and even memory cells, which means that even in future, if you decide to use the same donor, still the immune system of the recipient has a memory on their HLA, and they will reject that particular graft. So it means that even if you were to redo the transplant, you would have to look for another donor and repeat the whole transplant workup, ensure that the HLA matching is okay. And maybe that time, because this patient is already sensitized, you would go for a perfect match, which is rare to find. Maybe for identical twins, you might be able to find. In a family, if you type people in a family, HLA matching, you're likely to get 50%. That is haplotype, half match. But full match, 25% chance of getting. Remember, these genes are highly polymorphic. So even if you are identical twins, you will never have the same MHC molecule. So in direct allorecognition, as we have said here, the antigen has the recipient APCs has to engulf the donor halloantigens, and they have to be fully processed and then displayed on the surface of the MHC class 2, presented to the CD4 cells of the recipient. So alloreactive T cells in the recipient may be activated by both pathways, as we have seen, and these T cells will migrate into the graft. So once they've been activated, they will differentiate and they will become effector. Effector means they are now able to, they don't need like a secondary activation because they have a clear memory. Remember, the immune system is made in a way that in absence of the disease, everything stays balanced, like no reactions takes place. And in case something happened, then that's how now you end up developing autoimmune infection, because now you'll have the immune cells failing to recognize self. And while T cells are developing, there's that stage of selection. So in selection, what happens there? What the system just tries to see whether the T cells, only those T cells which are able to recognize the self MHC, those are the ones that will acquire the protective shields. And those are the ones which will be spared. The ones that are not able to recognize will be killed, and then they will die through apoptosis. So now in the lymphoid organs, there are the naive T cells. So those naive T cells can be activated by either direct pathway and indirect pathway. At this point of our lecture, we are now very much aware of how the direct happens and how the indirect happens. So it's the direct pathway is most important for cytotoxic T cells mediated acute rejection. So here that is now the CD8 positive cells, and then indirect pathway will play a greater role in chronic rejection. We are mostly the major type is the helper T cells, that is the CD4. So indirect pathway injuries that major responding T cells are the CD4, as I've said, and then these T cells could recognize the graft antigens displayed by the antigen presenting cells and secrete cytokines that injure the graft. Alloreactive CD4 positive, that is the helper T cells, will differentiate into cytokine producing effector cells that damage the graft, and the reaction will resemble that of delayed type hypersensitivity reaction. So as we have said, remember the helper CD4 is a very important element in immune system. Actually, any immunologist is so much interested in CD4. Why? Because with CD4, you are able to, if you present them with antigen, they are able to produce cytokines. They will stimulate, that stimulation will result into production of cytokines. You can have your interleukin 2. Interleukin 2 will stimulate the production of cytotoxic T cells like the CD8 cells. We have got now other cytokines like interleukin 5 and 4. Those ones will activate the immune response. That is the B cells. They will differentiate into plasma cells and give us antibodies. And then we also interleukin 10, which will recruit more macrophages to the site of infection. But now in this presentation, we're talking about graft rejection. So therefore, when alloreactive T cells are stimulated by the indirect pathway, the principal mechanism of rejection will be delayed type hypersensitivity mediated by the CD4 cells. And these CD4 cells will be able, that there's traffic, like they'll be able to infiltrate the graft and recognize the donor alloantigen. So once they recognize the alloantigen, they will mount and mediate or a very strong immune reactions that end up destroying the tissues of or the cells of the newly newly implanted tissue, or in this case, we are talking about a graft. So alloreactive, on the other hand, the CD8 cells are activated by the direct pathway, and then they differentiate into cytotoxic T lymphocytes. As you know, and we have mentioned, we have mentioned at the start that the CD8 positive cells, they'll be able to recognize the donor class 1 MHC molecules, and then by doing that, they get activated, they differentiate, they form effector cytotoxic T cells. What these cells does is that they will release things like perforins. Perforins will be able to create pores, to inject pores or things like gran enzymes. Those are serum proteases that are able to cleave these particular particular cells. So it could be endothelial cells or any allograft antigen. So now remember, all the cells of the allograft in the recipient system, they are referred to as an alloantigen. So you can imagine we are now having so much traffic of the cytotoxic T cells into the they are infiltrating the newly implanted graft tissues and mounting and creating. Remember, once a pause has been created to that, in a cell, what happens to it? It will die. It suffers the apoptosis. And then the CD8 positive cytotoxic lymphocytes, they are generated by the indirect pathway. Those ones which are generated by the indirect pathway are self-MHC restricted, and they cannot directly kill the foreign cells in the graft. So that one also is important, so that the immune system has it in a way that it's balanced, so that in the process that it is mounting a strong reaction against the foreign antigen, it doesn't end up killing the recipient own cells. So if that happens, then now that's how we'll end up having a graft versus host disease, and that is another whole area that we can discuss maybe in future presentation. And then the CD8 positive cytotoxic T cells induced by the indirect by direct recognition of alloantigens are most important for acute rejection of allograft, whereas those ones of so these ones we have explained, just a repetition of what we have said. So the diagram there now join the two. So it shows you the direct pathway and what happens. And now in a nutshell, everything, all these terminologies that we have said, has been summarized in this particular diagram. So if you look at, for example, the column for the direct pathway, you have got the antigen presenting cell of the graft interacting directly with the recipient cells, that the CD8 positive cells. And then now, once the CD8 positive cells have been activated, differentiate and give us the cytotoxic T cells, and then the cytotoxic T cells are able to mount well. And you can see, look at where the cursor is. You can see the cytotoxic T cells are able to recognize the cells, the endothelial cells lining the renal tubule, and mount an immune response, destroying these particular cells. And this is just a newly implanted graft. So you see this particular graft will not be able to survive the immune attack. So in other words, also, the cytotoxic T cells can also attack the blood vessels. That is, they will bind to the endothelial signs which are lining the blood vessels. And once that one happens, remember they cause injury to the endothelial cells, and that one will result into production of things like von Willebrand factors. So von Willebrand factors, what will it do? It will activate the platelets, resulting into platelet aggregation and even formation of clot inside this particular graft. So you can see how strong the immune rejection is, because now we are having the tissue will suffer both ischemic damage in that it's not able to receive blood, supply of blood via occlusion. The clots will occlude the vessels. And at the same time, we are having cells, the cytotoxic T cells, which are destroying the implanted cells directly. So that is how the organs will fail completely and finally removed, so that the symptoms can be relieved from the recipient. Remember, this is already a recipient with a failing kidney. And for a patient to reach that point of transplant, it means that both kidneys have failed. They cannot survive on one. And then, if you go up the diagram again, more of a, just where the direct pathway is, more of a semi-direct pathway. The same antigen presenting cells is engaging the CD4 cells. So what happens here, it means that the same antigen, the same dendritic cell that is in that diagram is able to phagocytose and process the alloantigen and present on MHC class 2. And then this will be recognized by the recipient CD4 cells and mount an immune response that is similar to the indirect pathway on the other hand, as you can see. And then that one will give us more of the CD4 cells. And then these helper CD4 cells are able to release cytokines such as interferon gamma, and also things like interleukin 10, you can see, which will activate more macrophages, and these macrophages will be able to attack the cells of the newly planted organ. And that one they can do through ADCC, that is antibody-dependent cytotoxicity, where they do this they cause injury to the implanted cells through release of, by help of lysoenzymes or proteases, things like that. And then they cause cytolytic activity on these particular cells. And then the same alpha CD4 cells can release things like interleukin 4 and 5 that recruit more of B lymphocytes. And remember, these B lymphocytes will differentiate into plasma cells, giving us antibodies that are very specific to the what? Very specific to this particular halo antigen. So we have here, we'll have an halo specific antibody. And you can see where my cursor is, we have got allospecific antibodies binding to the inside the blood vessels now of the newly planted graft. And then once they bind to that, they cause what? Again, damage on the endothelial lining of these blood vessels. And that exposure of the subendothelial lining will release, will activate these cells to secrete things like convoluted blood factor. And then now what happens? Platelet aggregation, and finally clot formation inside this particular tissue. So that is how the rejection takes place. So at least by the end of this presentation, we have a picture on what happens. That is the semi-direct pathway, but semi is I not really want, because it is just a bridge of the two. So mostly we'll be talking about the direct and indirect allorecognition. So the table there summarizes what the difference of the two. Like in direct allorecognition, here we are talking about intact allogeneic MHC, but now in direct allorecognition, they have to be processed. So antigenic peptides of allogenic MHC. And then in both, we are using, remember in direct, it is the the donor APC that is presenting the allogenic MHC. But in indirect allorecognition, we have got the APC of the recipient processing the halo antigen of the donor, and then presenting them on its MHC class 2. Then mostly it will be CD8. Actually, it is CD8 that for direct allorecognition, and then CD4 for indirect allorecognition. For the CD, because remember, this CD4 will still produce interleukin 2 and then the cytotoxic lymphocyte. But we have said those ones are MHC restricted, so that you don't end up injuring the these alloreactive cells do not end up injuring the recipient's own MHC. And then function in graft rejection, for direct, it will result into acute graft rejection, and then for indirect, chronic graft rejection. The degree is very strong for direct allorecognition, and it happens so soon, a week, within days to weeks. And then now for indirect allorecognition, it might take time, and sometimes it could be a progressive from the acute rejection. It can progress to chronic rejection, but mostly 1 year after transplant. So you can see, maybe for a transplant patient, they don't celebrate so soon. It is after a lot of follow-up and all that, and the new kidneys, the newly transplanted kidney or the organ is working very well. There is no signs of graft rejection and all that. And maybe these signs might not be there in the early days, only to appear a year later. So even after 1 year, they still have to be followed. And then again, remember, we have to really make the immune system of the recipients to be as tolerant as possible to accept this particular graft, so that allow time for and grafting until now the immune system of the recipient sees the newly implanted graft as its own and not foreign. So still these patients will be in long-term, maybe in long-term immunosuppression drugs or immunosuppression treatment. So on the sensitization, it becomes very easy now to talk about it because it's just a summary of what we have discussed. So maybe the next one we need to talk about it. So what happens? The antigen first is recognized by the T cells, and then that triggers the lymphocyte proliferation, following the antigen processing and presentation, as we have discussed, in direct allo recognition and in both direct and indirect allo recognition. So we also have minor histocompatibility complexes, which play a role. They don't need a match, but multiple mismatches can trigger rejection. So the minor histocompatibility complexes, they can also trigger a rejection in case where there is a lot of mismatches. But what we normally typed, or what will be of our interest, are the major histocompatibility complex, that is the HLA antigens. Minor histocompatibility complex mostly plays role in the innate immune system, things like natural killer cell. Yeah, those ones, they plays role in the mostly in the innate immune system. And then important now, the major histocompatibility complex can be recognized directly by T cells, while the minor ones, as you can see in the direct recognitions for the major HLA antigens, the donor APCs can interact directly with the recipient, the donor, let me say the donor MHC. Yes, the donor MHC can be able to interact directly with the recipient antigen presenting cell. That's not the case for the minor histocompatibility complex. So for the minor, it has to be processed. Yes, like what happens in the indirect allo recognition, and then presented. Then that's how the immune system immune. So in this picture, we have got a donor kidney. This is a kidney transplant, as you can see where the cursor is, that is the graft. So in the graft, we have got the donor dendritic cell and the recipient dendritic cells. Remember, once it has been anastomosed, it is a one system. So even the blood from the recipient will flow directly through the newly transplanted through the newly transplanted organ. So again, in that exchange, we are seeing the allograft, that is the donor dendritic cells, and also the recipient dendritic cells. And then where I'm pointing now, there is the donor alloantigen. This could be cells. So this recipient dendritic cells have already phagocytosed the donor alloantigen and processed it. And then now we have two people now moving to the secondary lymphoid organ of the recipient. So we have the green one, the one that is leading, that is now the donor dendritic cells displaying the donor or the foreign allo MHC. And then we have also the recipient APC with a processed allo MHC, displaying it on its surface. Then they move to the lymph node where this will be recognized by now the naive T cells. So we have got the recipient T cells. So naive T cells here, the CD8 cells will be able to interact directly with the, and here the CD8 cells are the one in blue in color. They'll be able to engage directly with the donor dendritic cell, that is the allo MHC. And then we also have the CD4 cells, which are in red. So the CD4 cells here, what they do, they interact with the displayed donor peptide MHC on the surface of MHC class 2. And then they get activated. So once activation has taken place, tumor growth, the growth factors are released, they differentiate, they proliferate, and they become many in number. So now we are having several of them, the recipient effector T cells, And these are the ones that are now ready to traffic back and mount a strong rejection on the graft. So in the effector stage, that's the stage 2. The host immune system attacks the graft, destroying it with 4 methods. One is the cytotoxicity, lymphocyte-mediated cytotoxicity, what we have seen in direct recognition, and then now we have got delayed type hypersensitivity that is happening in direct recognition, and then antibody-dependent cell-mediated cytotoxicity through more cytotoxic cells like the macrophages. They're also able to mount, even the eosinophil is part of the ADCC. And then complement-mediated lysis. Again, those antibodies are able to activate complement and result into the allospecific antibodies are able, they attach to the endothelial lining of the graft blood vessels and then activate the classical pathway of the complement system. And the end result is lysis of the cells. And finally, destruction. So now you can join the dot at the sensitization stage. And then this second picture, so you see now we have, the journey is now starting at the lymph nodes. We now have our effector cells. Remember these effector cells, both the CD4 and the CD8, they are already presented with the alloantigen and they are able to recognize this particular alloantigen. So they find their way to infiltrate the donor or the newly implanted tissue and then mount an attack on the cells. And then finally that one will lead to the death of the newly planted. And this is a summary of both the sensitization stage and the effector stage. Sensitization stage, I would not want to repeat, but exactly as we have said. So activation, and then now we have the effector T cells, they traffic, they infiltrate, and the rejection. Then they end up killing the cells of the newly implanted graft. So yes, it is true. Graft rejection is the monster to watch in organ transplant, and aim of the major fear or the major goal of any transplant is to minimize graft rejection as much as possible. Otherwise, you would have done a lot of work, but with no benefit to both the donor and the patient. Because remember, these donors, they are making a personal sacrifice. They are ready to, if it is a kidney donor, they'll be able to survive. They will sacrifice to survive on one kidney and then save the other person's life. Then we end up having rejection. So HLA typing is very key. I know it is a complex area, but it is very key that the donors must, if not fully matched, but at least we should have an haplotype match. And also, again, of course, the history of the recipient, whether there have been multiple transfused, and also you see when the cross match will still come in, because you really have to ensure that they don't have the blood, that is the blood of the donor and that of the recipient, are compatible. Yeah, so there's a lot of workups that, the transplant workup that will go on, and even the doctors will do a lot on their side to ensure that the incoming organ is, the recipient body is ready for the incoming organ. So you really have to suppress the T cells. We really have to block the interleukin-2 pathway so that we reduce the number of cytotoxic T cells that are released. Yeah, so they'll be on immunosuppression therapy. And as I said, this area is quite a huge topic. So maybe in the next presentation, we'll be able to look at how the effector mechanism of graft rejection look like. So in that case, we'll be looking at now, how does the acute rejection happens, how does it present, and then how the acute rejection phase and the chronic rejection phase. Then if we cover that maybe in the next presentation in future, then we'll have a clear picture. But it's more or less the same what we've discussed, because everything starts at this point. It has to be a allograft antigen being recognized by the donor immune cells. And then from there, other mechanisms comes in. We have more effector cells being produced, and they are destroying the newly graft, and then that one is rejected. So we are also looking for an optimistic future, hoping that we'll have more transplant center in Kenya and more such. The most important, because now as we are in the lab, maybe advancement in technology. I don't know whether we have a center where we can do HLA typing so that we don't have to be sending samples very far away. So that's another area to look at in terms of laboratory medicine. Are we able to do sequencing? Yeah, because maybe at Cavi they are doing the mixed lymphocyte reaction. I don't know, of course, maybe they do with them. I know they work with vaccine development and all that. And so there's a lot of immunology, there's HLA and all that. So maybe they are close to HLA typing. And if somebody has knowledge or a know-how where there is a center for HLA typing, you can share with us. That will be a great improvement for our nation. So thank you all. So the monster is graft rejection. So whenever you hear a transplant or anybody who requires transplant, the key thing is to avoid the graft rejection as much as possible. Then the new organ will be able to graft and pick up the function of the failing organ. That way we'll be able to relieve the patient of their symptoms and disease. Thank you. Back to you, Bruce.
So much, Daktari. It was a complex area. Looks so simple. It is an area that I think, if I remember quite well, it took us about a semester to cover, and you've made it simple in 1 hour. So thank you so much. And as have you said, these are large area, and we would really like you to prepare for us another presentation on the same. So if I can read from the people's comments, John Chomba is saying a repeat of this is necessary since it's a complex subject. Thank you, John Chomba. Jacqueline Kili is saying this is a great presentation. Jacqueline Simba is saying this is so wonderful, Daktari. Lina Todiambo saying a nice presentation. Jafar Abdullahi, a wonderful presentation. Ibrahim Bala, nice presentation. Edwin Keter, great presentation. Yeah, so General Daktari Francis, it was a very wonderful presentation, and really thank you for that. So straight, I'll go to the Q&A. So our first question is from Wilfred Aria. Wilfred Aria is asking, what makes to CD? Let me come up again. Wilfred Aria is asking, what makes alloreactive CD4 T cells respond more strongly to donor MHC than to foreign antigens like bacteria and viruses? Francis, if you can respond to that. Hello, Francis.
So remember at the introduction, we said MHC molecules are highly polymorphic, which means each and every gene subtype there has got several variants. If you look at, let's talk about HLA-B, it has almost the ones that have been typed, 350 alleles, greater than 110 alleles for that particular locus alone. So that's what ignite a very strong immune reactions with the donor, with the recipient lymphocytes, or rather immune cells. Yeah.
Thank you so much. Another question. From Wilfred Aria. Wilfred is asking, in transplantation, can Willebrand factor be a therapeutic target in transplantation? Daktari, can respond to that?
In transplantation, because now you are looking at how is this thing coming up? For the von Willebrand factor to be released, then it means these cells have been activated. So mostly the target of therapy will target the T cells. That is, if it is B cells, then you reduce the production of cytokines, things like interleukin-5 that ignites the production of these allospecific antibodies. If it is the cytotoxic CD4 cells, then we use molecules like tacrolimus. Those ones will inhibit that pathway of interleukin-2 production. So interleukin-2 is the one that stimulates production of the cytotoxic T cells. So what happens is that you try as much as possible to arrest the immune response, where the production of the suppression therapy is just reducing the production of aggressive lymphocytes in the recipient system. Yeah. Yes, Bruce.
Thank you so much, Dr. Jacqueline Simba is asking, is there any test that is normally ignored? Jacqueline Simba is asking, is there any test that is normally ignored that leads to tissue rejections, more so the autograft?
Okay, Bruce, you're able to hear me?
We are able to hear, but you're breaking.
How about now?
Yeah, we are not able to hear.
For autograft, you see you are using the same HLA, the recipient MHC molecules itself. So chances of rejection for autograft is very rare. So I can't pinpoint one test that is usually, because transplant is actually a very risky process. So you really want to, hello? Am I clear now? I'm saying chances of rejection in autograft is rare because you are using the same patient organ and then you transplant it. Maybe it is a liver, you cut a small part of the liver and transplant to the, because the liver is self-regenerating. So you can use the part that is not affected to arrest the diseased part. So in this case, because the system of the recipient is able to recognize self-MHC molecule, and chances of rejection will be very limited in autograft. Am I clear now? Hello, you are able to hear me?
Hello, Francis, can you hear me?
I can hear you.
Thank you so much.
Yes.
Francis Mambili is asking, what role does immunosuppressants play and how do they affect the health of a patient after transplant? What role does immunosuppressants play and how do they affect the health of a patient after transplant?
That's a very good question. So immunosuppressant drugs, what they do is they prepare the recipient to tolerate the incoming organ. So they will greatly reduce production of lymphocytes mostly. Most of the time they will target lymphocytes because, as you can see, most of these rejections are mediated by CD8 and the CD4 cells. Those are the major key players in graft rejection. For the hyperacute rejection, of course, we can do HLM. If the HLM matching is done, and we have a perfect match, and then also the cross match is done to ensure that the bloods are compatible, and also maybe we detect anti-HLA if maybe the patient has been sensitive. For those ones which are multiple transfused patients or maybe female patients who have had multiple miscarriages or something of that sort, and then now they end up producing anti-HLA. Then that one you will use drugs that are able to mop away the irregular antibodies. But most of the drugs in use will tend to repair the donor's immune system by suppressing their activity. So you suppress the activities of the lymphocyte as much as you can. So post-transplant, that's where the problem is. So these patients will become more susceptible to, because now they're incompetent. So they will become more susceptible to infections, things like cytomegalovirus, fungal infection. So those are the things that we're likely to see. So another thing is also cytomegalovirus free. So you have to look at those testing. Post-transplant, the doctors will make a follow-up with tests like cytomegalovirus. So any other opportunistic infections, like we see in immunocompromised patients, they are more susceptible because the immune system is already suppressed. I hope that one has answered the question correctly.
Thank you so much. Thank you. Another question is from Topiko Memia. Topiko is asking, can a donor organ reject a recipient, or it's always the recipient to reject the donor? Francis?
Yes, Topiko, thank you so much for that question. The donor organ can target. They don't reject now. You can't reject what is not yours. So with them, they come and mount target to the recipient host cells. And that is now another topic. It is graft versus host disease. So instead of the recipient immune cells mounting strong reactions to the donor cells, what happens now? The donor lymphocytes are the ones, and this is worse because the donor lymphocytes will traverse the I'm audible, Chris. Eh?
Yes, you are audible.
This is once because the donor lymphocytes will be able to traverse the system and attack the skin, attack the gastrointestinal system. Those are the most affected, and the liver. The three organs mostly are graft versus host disease are the most affected one. So it's not the donor recipients rejecting, but they are now mounting an attack to the recipient host cells, and that will result into graft versus host disease. Yeah, so in that case,
thank you so much.
Yeah. Okay.
Thank you so much, Daktari. There are several questions here. Don't know whether we'll exhaust all of them, but let's try. Topiko is also asking if CD4 is key in damage of graft.
Yes.
Does it mean HIV patients with low CD4 counts are in a better position to receive grafts?
We can't say the HIV patients are in a better position to receive grafts because one, they're immunocompromised. Yes, the goal of the amount of response they will elicit towards a graft is quite low, but they risk, in fact, chances are very high that they risk suffering from graft versus host disease, because what this one means is that the incoming lymphocytes are more competent or immunocompetent to the host immune cells. So it is the vice versa now.
Thank you so much. Another question from, another question. What is the recommended biological sample and timing for tacrolimus testing?
Recommended biological sample is whole blood. For the timing, if it is the timing, of course, the doctor will specify the time that we are interested in. But if you are for monitoring purposes, is normally 30 minutes before the next dose. Yeah, I think I'm correct on that.
Yeah, if I can add something, it's normally if it's before the dose, then it is at the trough level when its dosage is very low. Yeah. And we normally test trough level and peak levels. Okay. Another question is from Eric Awonda. Can you explain peripheral blood cytopenias? Okay. Can you explain peripheral blood cytopenias? in relation to BMT, that is bone marrow transplant. Cytopenias. Okay, can you explain peripheral blood cytopenias in relation to BMT, that is bone marrow transplant?
So in bone marrow transplant, if there's peripheral, okay, I'm assuming this patient has been transplanted, like we are now talking about post-transplant care. So if they have received the bone marrow transplant, we expect the new bone marrow to pick up. Of course, there will be time for homing. That is the new stem cells to adapt to the recipient microenvironment. And before that, there must be, even pre-transplant, I can say, because now before you do a bone marrow transplant, there has to be myoablation therapy. So what happens in myoablation therapy is that you do away with the deceased stem cells. If it is leukemia, for example, you remove as many stem cells as possible. So once the stem cells have been removed, it means that there is almost zero stem cell that is left to produce, to continue producing the cells that we see in the peripheral circulation. So in that case, we are having peripheral blood cytopenias. For the post-transplant care, if at all if the newly transplanted stem cells have not picked up again, and maybe there is also cases of graft versus host disease in BMT, then you'll also be able to see a case of peripheral cytopenias. It means that the bone marrow, the newly transplanted bone marrow, are not functioning optimally to continue producing cells and keep the normal level of blood cells, white cells, the platelets, and the platelets and the red blood cells. I hope that answers this question.
Thank you so much, Daktari. Another question is, can a rejected organ be given to another patient?
Oh, that is a disaster. Rejected organ already.
Okay.
Yeah, that is because remember this rejected, it is already has effector T cells, cytotoxic. There's so much infiltration. If you give it to another person, you are mounting the immune response immediately, almost immediately. Remember, as much as it is polymorphic, in the population, let's say African ethnic subgroups, the HLA, A, B, C for the class 1, the HLA, DP, DQ, DR for the class 2 is still there, and chances of even unrelated donors having almost the same MHC molecule as mine is there. So you can see it will be a disaster. Plus that organ alone, remember the reason why we are removing it is because it has suffered ischemic damage. It cannot function now. The cells have already necrosed, or they have already suffered necrosis, tissue cell death. So it is a dead organ, in short. It will not be able to pick in the new for the next recipient.
Thank you so much. If chronic means weak rejection, is it reversible or slowed or treated or stopped? If chronic means weak rejection, is it reversible? Is it slowed? Can it be treated or can it be stopped?
For chronic, it can be stopped by close monitoring, or rather, maybe the strength reduced. So what happens here? That's why these patients are in lifetime immunosuppression drugs. So if the doctor is seeing signs of chronic rejection, they might have to adjust the dosage of the immunosuppressive drugs and even use more other. So we'll now approach it from the physician point of view. They will just look at the management plan and see if it is working, and of course continue with the close monitoring and all that. Yeah.
Thank you so much. Martin Jugun is asking, how many HLA antigens that may be expressed on cell surface?
So HLA antigen, we've said we have got class 1 and class 2, and the class 1 has got 3 HLA antigen. Class A, MHC class 1, that we have got HLA A, HLA B, HLA C. And then now we have got class 2, that is HLA, DP, DQ, DR. So these 6 antigens will be expressed on the cell surface, and they are inherited like that as an haplotype. So you will get one group haplotype, one haplotype from the parent, having both the class 1 gene and class 2 gene, and the next one from another parent. So the problem is with the polymorphism, in that even in a family, chances of you getting a donor, HLA donor, in a family, that is the nuclear family I'm talking about, same mother, same father, siblings, is 25%. It's a very low chance. Because remember, what will be in my system could be DP60, and then my siblings having DP70. That's already a variant. Again, my sibling having an A10, me I'm having an A8, and mostly you will find if it is A, they are in pairs. Like A will have 8, 10. And then now B again 20, 30. Like that. For the DPQ, the same. For the HLSC, the W3, the W4, so it has got a lot of variability. So what you can easily find in a family is an haplotype match, where maybe 90% of the population, when in their family, nuclear family, they're able to find a sibling that is matching them half. So I hope that one answered the question, Bruce.
Thank you so much. There are several questions, but I'll only do 3. Another question is from Lilian Chenjeni. Lilian is asking, what is the first thing to do? What is the first thing to do in the case of a graft rejection?
So, like now you've said in the chronic rejection phase, because it is a week and maybe it is established a year later. So that phase, the doctor might adjust the dosage, or they change the management. Maybe if the regimen, maybe the patient was on tacrolimus, cyclosporin, and some, these other ones are called what NSAIDs, the likes of prednisolone. Yes. So those are the things the doctor will do for chronic rejection. But now for acute and hyperacute, these ones elicit very strong immune rejection, and would mean removing the implanted graft. Because for hyperacute, you can't save it. Remember, it's already the occlusion happens immediately in a matter of hours. The patient, the recipient already has the alloantibodies in their system that amount are very strong, that will attach to the endothelial cells and then result into clot or occlusion, a lot of plaques, platelet aggregation, plaques being formed inside the vessel, and then it suffers permanent ischemic damage. So even with the chronic, they will still have to reassess, because even with the chronic where there is arterial occlusion, maybe it is something that has developed over time. Then the arterioles are completely blocked in a way that you cannot, blood cannot pass through. So you see at that point, the tissue will die.
Thank you so much.
Yeah.
Thank you so much. Another question from Yona Kutete. Yona is asking, how is liquid tissue rejected?
Liquid tissue like blood. So in MH, in rejection, whether it is solid or liquid, we are talking about the HLA antigens. That's what mostly elicit the immune response that result into a graft rejection. So it is the same thing that we've discussed: the direct allorecognition, the indirect allorecognition, and the effector cells being produced, and then the amount immune response. Yeah. So if it is, we are talking about, let's say if it is stem cell transplant, then you will find that the system, these stem cells are not able to engraft themselves into the bone marrow microenvironment and start performing optimally. So they get rejected through that. But the recognition pattern is still the same thing, the MHC. The donor MHC that is in either the solid tissues or in the liquid tissues.
Thank you so much. The last question if donor APCs die off over time, how does the immune system sustain graft rejection?
The donor APCs dies, or the recipient APCs dies. Because for the graft rejection now, it is the recipient APCs that plays the key role. So the donor APCs, even if they die, they will still be engulfed by the recipient antigen-presenting cell phagocytosis, and those peptides presented on the surface of recipient APC, and the mount an immune response, and the final product will be a graft rejection.
Thank you so much. Maybe the last one, which I think is compelling, is David Ndirangu is asking, how do we handle HIV patients in transplantation? I don't know whether if the HIV patient is a donor or, but mostly the HIV patient is a recipient. So how do we handle HIV patients in transplantation? Daktari?
It will be a delicate balance, right? A tricky point for the doctor to make a decision of transplanting HIV patients. So one thing will be to establish the CD4 count for this particular patient. Because remember, they are already immunocompromised. So one is the viral load. What is the viral? In this case, we are not transplanting to cure HIV. We are transplanting just simply because this patient is HIV positive, and they also have, we call them what? They also have other chronic illnesses. They also have other chronic illnesses like renal failure, and maybe now they need transplant. So in that case, then the doctor really has to assess whether the HIV patient will be immunocompetent to accommodate. Because if that one is not assessed, then we'll have a graft versus host. Instead of the recipient immune system reacting against the donor, it will be now the donor lymphocyte destroying the recipient cells. And you would have not helped this patient, you would have worsened the case. So HIV patient, what is their CD4 count? What is their viral load? And what is the assurity that this patient will remain or adhere to the HIV medication so that they keep their immune system as stable as possible? So in such a case, I think the doctor might consider alternative therapy options like dialysis in such cases. Unless now the patient is responding, their CD4 counts are okay, almost back to the normal, then now we can, at least there is a reduced chance. Okay, it is a tricky balance because remember, still, even if the CD4 counts are okay, we still have to suppress those CD4 further. So to me, I would prefer alternative therapy for not just HIV, but for other patients which are immunocompromised.
Thank you so much, Francis. We've come to the end of the presentation. And before I give you time to give your final remarks, I would like to put across a few points. Number one, that this webinar will attract 3 CPD points that will be credited to your KMLT-TB portal within 24 hours from now. Then one of the acceptance criteria or how do you qualify for the CPD, 3 CPD points are that you need to have attended 60% of this webinar. Then the other announcement is that whenever you receive a communication from us, maybe there's a failed upload from our end, maybe there are, for whatever reasons, your details that you shared were incorrect, please respond immediately to our emails so that we can upload your points without failure. Because if we cannot, we are restricted to upload points beyond 2 weeks. So after 2 weeks from now, we'll not be able to upload points for this webinar. So please respond to our emails. Dr. Francis, thank you for your time. Maybe this is your chance to give your final remarks, then we end the webinar. Welcome.
Thank you, Bruce, and I would also love to thank the entire LMSK At the Nairobi Hospital, we have a transplant program. So maybe if you know someone that needs transplant, they can always come to our facility. We offer both pre-transplant care and post-transplant care. And it is a comprehensive program run by, if it is kidney, we have a very experienced and highly specialized nephrologists on board and other specialists from from different areas of healthcare. Another thing, we are looking to a future where most of these things can be done in Kenya, especially HLA typing. I know in this webinar we have policy makers, so ours is just to persuade them, maybe Cambridge or Kavi, those ones who are actively involved in immunology research and advanced immunology lab. That way we'll open more opportunities and even save time, because you can imagine now if we have an HLA, a center where we can do the HLA typing here in Kenya, it becomes very easy. So even the transplant at Kenyatta, Nairobi Hospital or Aga Khan, the patient will not wait for that long for their results. So it will really reduce the TAT of the whole thing. Otherwise, it was an eye opener, and this topic is a bit complicated, so it is a learning opportunity for all of us, both me and the attendees. And I hope that we have imparted knowledge and all of us have gained something. Thank you so much, Bruce.
Thank you so much, Francis. And to the rest of the team that attended this webinar, thank you so much. We really value your feedback. If you check on us online through official channels of communication, you can always give us your feedback so that we can improve the webinars. Otherwise, thank you so much, and let's meet on Monday from 3 p.m. to 4:30.